Okay, good morning, everyone. Welcome back to our session for the first session for today. So it's our great pleasure to have Professor Sandip Kaur here with us. So Sandip has a very interesting background. So I'll try to give you a brief overview of his the kind of work he's done over his career. So he started his PhD at the Indian Association for the Cultivation of Science in Calcutta, where he was thinking about mathematical modeling of pattern formation and so on, from which he moved on to do a PhD at Virginia Tech. Oh, sorry, no, postdoc. Well, PhD was at IISc, and then he moved on to do a postdoc at Virginia Tech with John Tyson, where he started becoming interested in the cell cycle, modeling the cell cycle. From there, he did a second postdoc at the German Cancer Research Center, where he was thinking about signaling pathways and the combination of cell cycle, signaling pathways, and so on. Since then, he started his own lab at IIT Bombay in the chemistry department, and there he's been thinking of the cell cycle, mainly the cell cycle, and very recently he's also started his own wet lab, where they are doing some really interesting experiments, very sophisticated experiments as well in his own lab, mostly related to the cell cycle. So we're very happy to have Sandip over here. Over to you.
Okay, thank you very much, Shaon and our Mohit for inviting me here. And at the outset, I like the idea that giving a little bit of background of the subject and then giving a research talk, because sometimes it took me some time to give my background of the subject. So this helps a lot. So what I tried in this part of the talk or this introductory talk is just to show you how cell cycle modeling started and the evolution of the whole thing, and where we are right now, and how we can employ this idea of the modeling into the real life, to cure something and to predict something out of these models, which makes sense. Okay, so just to give you a brief idea that which kind of length scales and time scales we are going to talk about. So this is something I like this diagram actually. Sometimes what happens is that if you are doing modeling, we often don't appreciate where the biology exactly lies, but the length scale that we are going to talk about, we are mostly in the eukaryotic cell cycle regime. And obviously, the timing of the cell cycle varies between bacteria to lower eukaryotes like budding yeast to mammalian cell cycles can be from hours to even to days also. It depends on which kind of cell lines or things you are talking about. So mostly we will be in and around these time scales and these length scales. Okay. Even just to this idea again, this is not my way of putting things. This was happened first time when I saw such kind of a discussion in a meeting, and I like the idea that how a mammalian cell normally functions. It is if you look into the thing, is basically, if I think very broadly, is there are some input signals, and then obviously something gets processed and we get some specific output. And different cell types, we know that they normally give us different kinds of output. Okay, and even for different phenomena at the phenotypic level. Now, if I try to think about cell as an information processing systems, it will be kind of pretty similar to a computer that we normally use as an information processing system. However, obviously, if I really try to brute force compare both of them, there are similarities and there are dissimilarities in various aspects. The most important part is that although computers, when we can exactly program them and try to function different, get different outputs. For cells also nowadays we do synthetic biology and we can actually manipulate things and we get very specific output. But in general, still those outputs were still a little bit sloppy, and out of that sloppiness also we get certain specific phenomena. Still we can get certain definitive output. Okay, so definitely just the way we have something hardwired within these computers, there are something also going on within these cell types which are kind of hardwired, and because of which a particular signal, when it comes in the form of an input signal, gets processed and we get something out. And one of such phenomena is cell cycle, okay, cell cycle duration. We can think like that. Now, how things are hardwired within a cell, okay? There were many interesting articles you can find, but one of the nice reviews that you can find written by John and Bella in and around 2010, you could easily see that there are different signaling pathways, and obviously they get translated through all these signals get translated through different hardwired mechanism, and eventually it turns out to be some kind of an output phenomenon. Okay, if we look them very carefully, and it was not that John and Bella did this first. I will say in this regards, this motif things first was conceptualized by Uri Alon. There's a very nice book written by him also, and where you can find out that there are small motifs. The idea that they proposed is that there are small network motifs that ultimately allows these signals to process, and eventually we get certain outputs. Okay. Now, in this case, what if we start about some two-component systems, we can see that there are simple transducer process. We can have inverters. We can have negative feedback loops, positive feedback loops, and like double negative, which is eventually also a positive feedback loop. So different network motifs are there, and we can find out these network motifs very occurring at different places in these hardwares often, and they somehow couple them. Sometimes they interact among each other and give rise to certain specific phenomena. Okay, we will see that we kind of know that if we have say for example a negative feedback loop, we either can get oscillation. If there are other requirements also, not only that the presence of negative feedback can give me oscillation, it can also give me homeostasis. But if there are proper balancing of time scales, if I have a certain amount of nonlinearity in the system, eventually I can have oscillations also. So any oscillatory phenomena kind of has a negative feedback loop operative within the system. Similarly, if I have a switching kind of a behavior, that a cell state switches from a particular state to another state, we could think of there might be some kind of a positive feedback phenomena operative within that system. So that's the way they try to put forward the whole ideology, that there are certain network motifs and they functions in a very specific manner to give us some kind of a specific, allows the cells to give some specific output. Okay. Eventually, we can get to even more complicated, say, three-dimensional motifs, or like three-component motifs, and those are kind of either a coherent feed-forward loop or incoherent feed-forward loop. Essentially, if I start from a particular node, I can see that X activates Z in both direction, or maybe X inhibits Z in twice, so that again it becomes kind of an activation. So like coherently, it is kind of activating Z in these loops, in all of them. And if you look into any of these structure, they kind of incoherently activating Z. So basically in one direction it is activating, in another direction it is inhibiting. So these components also you will often see recurring I can easily figure out at least two of them from this picture. And if I really have the whole, all interactions within our cellular systems, we will be able to figure out different such motifs. So why I'm talking about these motifs before the cell cycle regulation? Because if you look at the cell cycle regulatory network, which is obviously a very complicated network, we can imagine that the whole process is tightly bound, and as we go from budding yeast to fission yeast to mammalian cells, there are similar proteins and genes which are actually coherently working together to give rise to a cell cycle phenomena. And we will see that there are recurrence of these feedback motifs. And as soon as I see that feedback motifs, I know for what they are existing within that network. So before going into the actual or diving into the actual cell cycle network motifs, we need to understand that why I'm at all seeing this phenomena. So it will be not a surprising thing for us. Okay. So just to what is the take home for up to this part is that there are certain motifs, they have certain roles to play. If I have a positive feedback motif, I can think of that cells are using that for some kind of a switching phenomena. If I have a negative feedback motif, they can be used for having that oscillatory phenomena within that cell cycle thing, and so on and so forth. And if I have some kind of this kind of a thing, so I can adapt to a particular situation, I can do certain specific things. So these are for adaptive response and other responses. So these network motifs has their own rule, and if you go to this wonderful paper written by Uri Allen, and I will also suggest to go to Uri Allen's book. It was an excellent book, so to read and understand these network motifs. Okay. So now actually we are kind of in the way to understand eukaryotic cell cycle. This is a very generic picture which I have drawn. So cell cycle normally has 4 different phases. So the main phase is the synthetic phase, where S phase, where the DNA get duplicated, and in M phase we know that whatever the duplicated DNA gets very faithfully segregated among the 2 daughter cells. And these are the very 2 important gap phases, G1 and G2 phase, especially the G1 phase. And there lies a very important point at this G1 phase, just before the G1S transition point, which is called the restriction point control. And most of the mammalian cells, and during the cancer progression, it has been found that this restriction point control mechanism is somehow faulty. What is this control does? So most of our body right now are alive, but not all of them are in the active cell cycle. Only some of these cells are doing active cell cycle continuously. However, if, say for example, some of my skin cells are now maybe dividing, most of them are not dividing. But if I have a cut, immediately signals will go, and the cells in the periphery will understand that I have to grow and somehow control the damage that has been occurred because of the tissue or whatever tissue damage that has happened. So basically those cells which were kind of alive but not actively in cell cycle will commit for the cell cycle. So right now these cells, most of these cells are in the G1 phase of the cell cycle looking for active signals from outside. As soon as they figured out the signal is above a certain threshold, they will opt for the cell cycle, and that has been tightly controlled by at this point, which is called the restriction point. Okay, and just not to go into superlative details, there is one protein called, or one set of transcription factor which is called E2F, and these transcription factors are extremely important because before the G1 phase they are at a very low level, they are maintained at a very low level. However, as soon as you cross this restriction point, the restriction point crossing is being designated by the high amount of sudden transition from the low level of E2F to high level of E2F. And recent experiments have shown, actually, not even recent, I will say at least 10-14 years before, the experiments have shown that so there is a control mechanism, and that happens in a switching manner. So the activation of E2F happens in a switching manner, not in a continuous manner or not in a graded manner. It just jumps from a low steady state to a high level of E2F just in a switching manner, which is switches. Okay. So this tells you that cells uses different kinds of motifs that we talked about just in the previous slides to have these sort of very interesting generic responses. Okay. Now, if you look into the little bit of more details, so there are certain proteins called cyclin-dependent kinases. Kinase means, I'm pretty sure, those who phosphorylates other proteins in different sites. Mainly these are tyrosine and threonine kind of phosphorylations that we are talking about in cell cycle here. And these cyclin-dependent kinases, as we understand, that they need a cycling partner. And these cycling partner comes in different phases at different point with different names. So at the very beginning you will see cyclin D, then after a while you will find cyclin E, then you will find cyclin A in the S phase, then you will find cyclin B, and so on and so forth. And they come in a very cyclic manner. And because of that, what you can observe, you don't need to know the, so this is a very interesting, nice figure that the video I got from the YouTube, this kind of a level free kind of a thing, and you can easily see that how the cells are dividing, and this is going on, so on, so forth, in a very cyclic manner. So essentially what it tells you that all these cyclines has to come up in a very temporal manner to say that right now is the end of G1 phase, right now is the start of S phase, right now is the start of G2M, then it should be M phase. And all these cyclines appear in a such temporal manner and a very sequential manner that it tells you that at the end, when the cyclin B is going down and roughly it reaches around half, the cell divides. That's the more or less, that's the way, sorry, there's a awkward music here also. That's the temporal manner, the way it happens. Obviously, there are other proteins which are also cycling along with time, and people have also captured how this, with single cell, reporters, people have seen that cyclin E is oscillating or something. But so you can basically look at into greater details of how these protein cycles. However, the important part is that because these proteins are cycling, eventually I will have a cell cycle phenomena. So cell will grow, and then eventually they reach at the end of the mitotic phase and they will divide. And this will happen time and time again, okay? Now, this is all these proteins, and obviously because you have activators, so there are many activators of the cell cycle, so there lies many inhibitors of the cell cycle also. For example, initially E2F is very low because E2F is held by a protein called retinoblastoma protein, which makes a complex with E2F, and it does not allow E2F to come out of it. So obviously the cyclin D comes up and phosphorylates RB protein partially, then E2F comes up and activates its own transcription positively, and then activates the formation of cyclin E, and then cyclin E further phosphorylates RB, and then E2F completely gets released, and eventually I will have a G1 to S transition. And that's the way, so on and so forth, cell cycle also progress. So I will come to that later, but the mechanisms are more or less well established in different phases of the cell cycle and why these things are happening. However, that was not the case when the cell cycle modeling started, okay? When the first time the cell cycle, one of the most well-known cell cycle model, and you can see this year, 1991, and you have to remember at that time, only cyclins were just discovered. People came to know about cyclins. Okay, these are the important proteins, and they regulate at that point of time. And I think in and around that time, they got Nobel Prize for discovering cyclins and CDKs and all these things. And this was kind of one of the very primitive and the first example of a cell cycle model. And again, I have given these two words, like what is called a phenomenological model and a data-driven model. Nowadays, what we normally do, it's a data-driven model. We get the data, we find a model, and then we try to understand the underlying kinetic parameters and other things from those by fitting the data with this, and we get a exclusive data-driven model out of everything. But at that point of time, that much data were not available. So what happens is that most of these models that was developed at that era, around 1990s, are phenomenological model. What do you mean by phenomenological model? We mean here is that I have a phenomena. For example, this model, what John was trying to figure out is for the frog embryo, what is the mitosis, rate of mitosis in frog eggs. And it was around, it is known that is about 30 minutes of duration. So the it's an oscillatory phenomena with a 30 minutes duration. And some of these underlying mechanism of the frog egg during the mitosis is known. For example, this CDC2 is nothing but a cyclin-dependent kinase in the frog egg. Only the name of the genes are different. And obviously they need a cyclin partner, and when CDC2 gets phosphorylated, and then only it can bind with cyclin and form a particular complex where cyclin also get phosphorylated, and eventually I need to get rid of this phosphorylation to make this complex active. This is that complex which is called a metaphase promoting complex, And you can see that there is a dotted arrow here. It means that somehow this metaphase promoting complex can activate its own activation. So kind of a positive feedback. And then eventually from here, cyclin gets degraded and I get back my CDC2. So there are two different things here. One, positive feedback loop, which is basically here. And there is a degradation which happens in a negative feedback format, although this picture does not show you that. And eventually, if you have a positive feedback kind of a thing, as I said, it's a kind, I can get a switch-like thing, some kind of a switch. And if I have a negative feedback associated with a positive feedback, essentially I can get a very robust oscillation. So what do we mean by that? Okay. So as I said, any say X is basically activating say Y and Y is activating X, something like this is there. I will say this is a positive feedback. And say I have some kind of a source, I can activate the source. If I try to plot the steady state dynamics of X as a function of S, I should get some picture like this. If I don't have any other things, I can obviously, I can model this. I can eventually produce a dynamics like this, where I have a stable steady state, which is a solid line, and I have another stable steady state, which is another solid line, separated by an unstable steady state. And this means that for a particular signal strength, I can have these state and these state accessible to the system. I can either be stuck in a low steady state of X or a high steady state of X. And if I somehow, and say this is my non-mitotic state, and this is my mitotic state, and eventually as the signal progress, I should jump and to my mitotic state. Now it could have happened in a very gradual manner, or it could have happened in a switching manner. Now how could I write at that point of time? They didn't know that whether it's really happening in a switching manner or not. However, the model eventually, if you do the bifurcation analysis or something, like if you try to figure out how the steady state of X is changing or as a function of signal, you will eventually see that it is happening in a switching manner at that point of time. So basically it's some kind of a model prediction that might be, it is switching. And then if you have something like this, that it activates something Z, and Z basically, you know, but basically it will. So basically that is true. No, 2 negative is also fine. 2 negative is also a positive feedback, and 2 positive is also a positive feedback. So X is activating Y and Y is activating X. And they will both keep on growing, but eventually, say for example, if I activate something, in this case it looks little like that. Even if you think like this, and then Z, maybe I have not taken the right example. In this case, what happens is that Z now will activate Y, okay? So that Y will again dominate over X. So what happens in the lower steady state is that if I have something like this, that X is now low and Y is dominating at this phase. And then eventually, because of enough signal, X will come up, and then I will switch to this point, and then X is also by that time activating Z, which will activate Y, and Y will again degrade X. So I should have some kind of a negative feedback operative in the system. So how that comes in, normally, if I have a normally negative feedback, what happens is that I have a stable steady state, and then it becomes unstable at a point called Hop bifurcation, some kind of a bifurcation where it becomes unstable, and I can have an oscillatory phenomenon. Now, if this thing is coupled with this positive feedback, what happens is that eventually my positive, because of that, I will have a Hop bifurcation point somewhere here, say assume, and I have a saddle node somewhere here. So if that Hop bifurcation point away goes outside this saddle node, then I will have a till here I will get stuck. But if I give this signal strength, I will oscillate. Okay. However, if my positive feedback is very strong, what happens is that put this Hop bifurcation point somewhere inside the saddle node of this. And then there is a beautiful phenomena happens. So what happens is that if the Hop bifurcation point was here, I will normally get a small amplitude oscillation. I will start, and then my amplitude also start growing, and my periods will also start growing simultaneously. However, at this point, if the Hop bifurcation point is somewhere here, I get a bifurcation called a saddle node invariant cycle. What happens is that at that saddle node point, I get an infinite period limit cycle. So there is a significance of that. So the significance is, normally in cell cycle, we had high amplitude oscillations, not small amplitude oscillations. And if the oscillation starts only because of the Hop bifurcation, just merely appearing here, and then I have this, and my oscillation starts here, I will still get an oscillation, okay? But my period will start varying very quickly. So say I start with 10 minutes, then it will immediately become 20 minutes, 30 minutes, 50 minutes. So that exactly I don't want. The other thing is that I want a high amplitude oscillation where the period will not vary much. And that can only be achieved if I have this kind of a bifurcation. And this bifurcation can only be achieved if your positive feedback is very strong, such that it allows the Hop bifurcation point coming inside somewhere here instead of, so inside the saddle node point, and then I will have this. So I will have a high amplitude oscillation, and my period will also not change with, if I change my signal strength. So normally in mammalian cells, if I try to model, so Tyson was a strong believer of this saddle node invariant cycle bifurcation, because I will have always a high amplitude oscillation, and my periods will also not vary if I change my growth factor. Normally what we see in, say, mammalian cells, for example, in our experiments also, if I give a 2% growth factor or 10% growth factor to HeLa cells, for example, my period will change from 23 hours to 25 hours or 26 hours, something like that. So the change in period is marginal, not that great, right? But if it would have happened that the change in period is quite big, like 23 to say 35, then I need something like a Hop bifurcation phenomena. But then my protein amplitude should also change like that. The amplitudes will start with a small amplitude oscillation, then it will grow into a big amplitude oscillation. But exactly that is what we don't want, because we have seen in experiments that the protein amplitude goes up rapidly high, but the period does not vary with the change in the growth factor. So that's the way they although at that point of time, I don't think even Tyson started thinking like that. Eventually he got that saddle node invariant circle because his positive feedback was quite strong, and because the positive feedback was strong, that makes your negative feedback loop very strong, and then the oscillation becomes quite strong. Make sense? If I have a normal Hop bifurcation, I will have an oscillation, but it will be small amplitude oscillation with changing period. So students, like, any question? Yeah. Frequency will automatically, it will automatically come. So as I said, frequency of oscillation is basically what I'm trying to say as the period of oscillation. So the period of oscillation, if I have a Hop bifurcation, I will see, for example, if I have a Hop bifurcation, I can also create 30 minutes. If I have saddle node invariant cycle, I can also create 30 minutes. Now, in both the cases, I can create a 30 minutes oscillation. That's not a problem. If you give us this, I can routinely make you a model. Does not matter. Now, the question is, what I see in experiment. I see in experiments that the protein level, when they oscillate, they go through via very high amplitude oscillation. But if I create a Hop bifurcation, see, initially the nature of Hop bifurcation creation is that I create first a small amplitude oscillation, then I move on to high amplitude oscillation, and then I go to very high amplitude. Then obviously again, if I have a Hop bifurcation point, I will have another Hop bifurcation point where the point ends. So it will again come down to a low amplitude oscillation. Kind of. And at that point of time, there is no such experiment, nothing is, it's like all these are, what can I say, in Hindi, what we say, a khayali pulao. At that point of time, it is all imagination in 1991. Right now, it's not an imagination. Right now, we see that there are high amplitude oscillations in cell cycle. Okay. When it was made, it was not like that. Okay. When this was conceptualized. So eventually what happens is that I write down my equation. Most of these are simple mass action kinetics, like what we use in chemical kinetics. However, there are some terms which are quite complicated. which just were given to reproduce the phenomena that I have a 30 minutes oscillation. Okay, so which we see here somewhere. So this is basically the positive feedback interaction, and the CT is nothing but the total cyclin concentration. Okay, we don't need to dive into the superlative details of the model. The idea is that eventually he did a 2-parameter bifurcation to figure out where is the region of oscillation or the robust oscillation, and he figured out that oscillatory periods of 30 minutes with reasonable high amplitude of oscillation. So that was kind of a model prediction that how mitosis happened in frog eggs. And when Tyson went to a conference to deliver this lecture, all those biologist people who are from who discovered the cyclins and everything, they said that you think a lot, nothing like this happens in real world. Okay, you think too much. And he was kind of he was very sad. That is what he said. Although he is right now smiling, he was at that point of time very sad after hearing this. Okay, and eventually he came back home disappointed, and then met this person who is Bela Novak around 1993 and 1992 basically in Virginia Tech, and they started talking about the same model, and they said, okay, let's make it a little bit more foolproof and more more robust. And this is basically the same model what they have proposed. It talks about the mitosis in frog egg extract. Earlier I was saying in the frog egg. Now I can take a xenopas extract, and same phenomena I can do it in experiments by doing that, although they are not doing that. So assuming that if I take a frog xenopus egg extract and I look into dive deep into the model, what I will be able to formulate is that I have still that CDC whatever cyclin dependent kinase. These are the 2 phosphorylation sites, and I have my cyclin. They can form a complex. This is an inactive complex, and eventually there is a protein called V1 which can phosphorylate and inactivate that complex. And there is a kind of a phosphatase which is called CDC25, which is another protein, which can dephosphorylate it and make it in a dephosphorylated state. And there are other phosphorylation and dephosphorylation partners. And eventually this is my active form of that metaphase promoting complex. And if you look basically this picture in little bit of greater details, what happens is that if I have unreplicated DNA, so I should get stopped at that G2M checkpoint. And what happens is that unreplicated DNA deactivates the CDC25, which was the phosphatase, which basically dephosphorylates it into the active form. So I will not be able to progress into the G2M. And this active form actually activates the formation of the CDC25 into the active form. So this is a double positive kind of a feedback. And similarly, I have a V1 and V1 phosphorylated. So unreplicated DNA activates the V1, and this activated complex inactivates the V1. So it's a double negative kind of a thing, because V1 also inactivates the MPF complex. So there is a double negative positive feedback loop. There's a double positive positive feedback loop. So I should have a switching kind of a phenomena, because I have 2 switches. But I like 2 positive feedbacks to control 1 switch. And there is a negative feedback loop where this active MPF complex activates the enzyme which activates the ubiquitin ligases, which degrades the MPF complex. So essentially I have a negative feedback in conjunction with a positive feedback. So I should be able to get a robust thing. So what they have figured out to begin with is like, although there are so many variables and everything, so it was kind of hard to foresee that what is going to happen. So they somehow make some steady state approximation on other variables which are not important, and they converted that whole system into a 2-variable kind of a thing, where I have the equation for this active complex, which is active MPF, and say for example I'm changing my total cyclin. So I have an equation for the total cyclin protein and I have an equation for an active MPF and nothing else. And they obviously they got those parametric condition where I will have such kind of a null line for active MPF. So if I put active MPF equals to 0, I can plot that graph, and this looks like something like this. And now you can see for different concentration of active cyclin, I can have only one steady state, one steady state, one steady state, but here I will have a 3 steady states, which are 2 stable and 1 unstable, and then again I will have another. So basically I can have a switching kind of a phenomena. So I have 2 steady states also sometimes, and then I will reach the higher steady state of active cyclin B or active MPF. What happens is that I can control this, the going up and down of this by changing different protein concentration. So no wonder that these are the inhibitors that inhibit INH or unreplicated DNA will switch this switch from towards higher side, and if I increase the form of CDC25, which is an activator, it will bring down that thing, which is fine. The other thing that what they have figured out is that if now, in addition to the switching phenomena, I have this negative feedback loop, I should be able to get some kind of an oscillatory phenomena around these two nuclei, and essentially they showed that they can get roughly about 80-minute oscillation that one can observe in xenopus oocytes for the M phase. That is what they showed. Now these are all model predictions, and again obviously this got highly criticized again in 1993. Who will believe without anything, without experiments? And believe me or not, Tyson kept probing on. I'm not praising him because he is my supervisor. What I'm trying to say is that he believed in his model. He tried to figure out a person who will do that experiment, who will satisfy this. And the prediction that model essentially made is that if I plot active CDC or MPF as a function of total cyclin, I should be able to get a low, high, and at that point it should jump. And if I can somehow bring down the total cyclin level, I should come down here, but I should not come down here, but I should come down somewhere here. So I should be able to see an hysteresis kind of a phenomena. And if I can somehow show that there is an hysteresis kind of a phenomena, that proves that there lies a switch. Okay, that was his whole intention. And who will do this experiment? So this experiment was extremely difficult, and I completely forgot the whole principle, but I will still try to explain. So what they have done, they collected the xenopus egg extracts, okay? And say for example, just look into this, that I have that extract at zero time point. I see that the nuclear envelope is completely, closed, nothing is there. And I can see even at the 90th minute also, the envelope is completely closed. So there is no condensation of the, DNA, okay, the chromosomes. However, around here you can see that the nuclear envelope is broken somewhere, and at some point of time I should have a condensed DNA. So this is my mitotic phase. So what they have to figure out is that I need to change my total cyclin level. But this xenopus egg extract has everything, the whole machinery to complete the M phase. So first what I need to do is to freeze the system, that nothing will change. I got the xenopus egg extract with everything, all the machinery, V1, CDC25, everything is there. But I will stop the protein synthesis by giving cyclohexamide. As soon as I give cyclohexamide, I stop any synthesis of the protein. I cannot stop the degradation of the proteins, but I can stop the synthesis of the protein. And they made a non-degradable cyclin, which cannot be degraded. Okay? And now they keep on changing that non-degradable cyclin and looked at the 0th minute and the 90th minute. Okay? And they see around 32 to 40 nanomolar of that non-degradable cyclin, you see that the nuclear gets condensed. So I can see this switch. Okay? And that is the first step. Fine. Can I see this? How I bring it down? To bring it down, what they have done, say I again had the xenopus egg extract, and at 60th minute and 140th minute, I'm just trying to look at, I can see that already the nuclear got condensed and everything is fine at the 0th minute, and also around 140 minutes roughly it is kind of similar. Okay. Now I start with very high concentration of this starting at 60th minute. So I start at 60th minute, and now at that 60th minute time point, I employ cyclohexamide. not at the time 0. At the 60th minute, I gave the cyclohexamide and also this 40 nanomolar of non-degradable cyclin. Now, when I look at 140 after 2 hours, I still see that that is again condensed state. So nothing is happening. So it is still in the upper steady state. And now they keep on repeating that, and around 16 to 24 nanomolar, you can see that here I start with the condensed DNA, but now my nuclear envelope is completely formed. So that essentially tells you that I jumped from here to there. And that's how they, for the first time, they proved after 10 years that there lies a switch. So you should believe mathematical model at the end of the day. And yes, please. What? That is nowadays you don't have to wait for 10 years. See, for example, Jill joined around 1998. So before that, the Tyson has no choice. Then to convince her took her another 3 years. So 2001. Then from 2001 to 2002, they did the experiment and they published this. And by the way, there's a very interesting thing. 2003 February. Not normally don't we write that. Why I have written? There is a reason. Not only Tyson believed his idea. Someone else also believed Tyson's paper. And they have, I am not going into these because these are superlative details. And what they have shown, Professor James Farrell group, they have shown exactly the same experiment with another non-degradable form of the cyclin B. But they have shown the same switch, everything. Like whatever they got in 1 month's time. It came out in Nature Cell Biology. So simultaneously 2 papers proving the same hypothesis came out in 1 month's interval in 2 different papers. However, they proved the same thing. So someone else also believed. So moral of the story is that if you make a model, if it is good enough and you have already a reputation, someone else will believe you and do the experiment. So yeah. So not only that, always you have to do the experiment. So Promenading is already also, he is a faculty right now. They believe this and they have the same experiments with only a different form of a non-degradable cyclin. Okay, great. Not only that, they believed further. So John was not allowed or not been able to convince Jill, because Jill was actually too much stressed out with that paper itself, to go further and prove the oscillator. However, James Farrell's group has done that too. What I said, so these are my positive feedback loops and this is my negative feedback loop. As I said, if I have a positive feedback loop, merged with a negative feedback loop, I can make my negative feedback loop really strong. If you look at the title of the paper, System Level Dissection of the Cell Cycle Oscillator, bypassing positive feedback produced damped oscillation. So the idea here is the following. If I go on weakening my positive feedback loop, I should not get my oscillator very strong and I should lose my oscillation after a while. That is what they are trying to showcase in this paper. So how they have done it? They first, take a different route. Okay, they first showed that they start with the model. They have their own model. However, the F factor, whatever this F means how strong is these 2 positive feedback loops are. They keep on weakening the positive feedback loop and you can see that the oscillator start losing its charm and eventually there is no oscillation. That's the model was predicting, that if I weakening my positive feedback loop, I will lose my oscillation. That is what it's saying. Okay. Now what they have performed in the experiment, obviously I am not saying that they first did so these are the way of saying things, that I did my model and then I see it in my experiment. Normally, I'm pretty sure they did the experiment first, they got it nicely and then they showcased it like this. Again, there is nothing wrong in it. The point what they have done is that they figured out what is the cyclin levels, for example, and they also figured out the kinase activity of the CDC2 kinase simultaneously in every 2 minutes interval, and they have plotted in this phase plot. And you can see that they get a 6.6-fold of change in the cyclin levels, and also I get a very beautiful oscillator. Okay, now and you can see initially the cyclin level comes up, and then only the kinase activity comes up, and then cyclin level starts to go down, and then immediately the kinase activity goes down. That's the way the oscillation should happen. So the positive feedback kicks in, oscillator kicks in, and then it comes down. Now, how to weaken the positive? In model, so very easy. I change one parameter f and I weakening my positive feedback. But experiment does not work like that. So for their experiment, what they have done, they first mutated these two, the tyrosine Y and T with A and F. I forgot, something is alanine, F I maybe proline, I guess. I forgot, whatever A and F is, they point mutated that Y and T. Okay, so now CDC25 cannot phosphorylate cyclin dependent kinase and activate it. So that positive feedback is weakened. And what they have done, think about this experiment. I don't know how to do it. They have point mutated all the 11 phosphorylation sites of V1, so that V1 also cannot get involved in the positive feedback. And now you see the fate of the oscillator from here to here. So you can see you came from here to here by doing experiment also. So positive feedback in coordination of the negative feedback gives you a robust oscillation in cell cycle, is kind of for the first time got proved. It took 12 or 13 or 14 years to prove it. Okay, great. And these are for frog egg extract. So who cares about frogs? So now we come into the real life scenario about our own mammalian cell cycle. And here is kind of the first mammalian cell cycle big model. And the only point of highlighting this is that here in this model, they have concentrated on this RB E2F kind of a regulation. That RB makes E2F in a complex form and does not allow E2F to go out. And because E2F cannot go out from that complex, it cannot get activated, I don't have my G1 to S transition. And eventually what happens is that serum comes in, they activate the cyclin D. Cyclin D phosphorylates RB in one of the positions, and then E2F gets partially released. Then E2F activates its own transcription. E2F activates the transcription of the cyclin E. Then cyclin E phosphorylates RB in another position. Then E2F gets completely released, and then only E2F comes up. However, in their model, they propose that E2F also comes up in a switch-like manner in 2004. It took another 4 years to prove this experimentally, and that was proved by Guang Yao. I forgot to get the picture of his supervisor. He was at that time, the PhD student. He's of my age only, and they did this excellent experiment. And I really want to show you, because this is what we are also trying to do right now in our lab, something similar. So this is basically the network they talked about only. Okay, so growth factor activates cyclin D, it phosphorylates RB, then E2F gets released, then E2F activates its own transcription, E2F activates cyclin E, it again deactivates RB, and then E2F gets completely released. So there are two positive feedbacks there. E2F activates its own transcription, E2F activates cyclin E, and then cyclin E activates this. So these are kind of a mutual activation. Okay. Now, what is they have done? They wanted to see the transcriptional activation of E2F. So they made a transcriptional reporter with D2 GFP. So this is a destabilized GFP. So I'm not a biologist, I'm just trying to explain it to the best of my capabilities. So this GFP is a short half-life GFP because E2F also have a certain short half-life. Now I need a transcriptional reporter of E2F. So it should show a dynamics like mRNA of E2F. So I have those promoter sites where E2F will come and activates its transcription of the D2 GFP. And I have the D2GFP 3' E2F region, so that E2F also, D2GFP also degrades at the similar rate of E2F. So that I can see the transcriptional activation of E2F. Now what experiments they have done? So the idea is the following. So they made a model, obviously, just to highlight, if E2F activation goes like, continuously, so I will have a continuous activation of the serum, and I will have a continuous activation of the E2F. However, the model, the thing is here is that I can think of that it does not happen like this, it happens like a switching manner. So I can say that the lower level of E2F means a G1 like phase, and higher level of E2F means a SG2M like phase. Okay, and if I now do the same experiments of changing the serum level, I will see at this point my E2F level will suddenly go up and remain in the upper steady state. And if I have some way of reducing my serum level from the upper steady state and I try to bring it down, I should bring down the level at this point, not at this point. So I should be able to see the hysteresis. Now, how I see this in experiments? What does it mean? It means that this is my bistable domain. So like I should have a population of cells where some of the cells will belong to low level of E2F and some of the cells should belong to the high level of E2F. That is the moral of the story is. So essentially, I should be able to see such kind of distribution. What does it mean? Like some of the E2F levels are, so in this axis, I have the, different cells with different concentrations of E2F, and some of the cells are having low concentration and some of the cells are having high concentration. Something like that, okay? So how to achieve that experimentally? To achieve that experimentally, they devised a beautiful experiments, which right now we are also doing routinely in our lab. So the idea is the following. First, I will go just going up, up, up. Like I just increase my different level of serum concentration. I will wait for 24 hours and check the E2F levels. Okay. And what happens is that if I go on increasing my, if I go on increasing my serum concentration, eventually if you just follow the red lines, you see that slowly a small bumps comes in. Why? Because I'm changing, but around here, I see there is some population, a high level of population, but there will be some small higher level population of cells also. So that's why these small bumps comes in, and then I will have a higher bump. So somewhere I'm somewhere here, and then I will have a smaller bump, then somewhere I'm here and somewhere here, and eventually I will lose and I will have a higher bump only. So basically, the distribution will tell me whether I have a unimodal population or a bimodal population, or again a unimodal population. That is what I expect. Now, if I go up like this, now how I come down? To come down, they set a beautiful protocol. What they did is that they gave a serum pulse. So initially you can see that I go up to 3% or 5% or 8% or something. So these are REF52 cell lines, so like rat embryonic fibroblast or something, primary cells. So what they have done, they have given a 20% growth factor like for 5 hours. Now how they know 5 hours? Again, by doing experiments time and time again, they figured out if they give 20% for 5 hours, cell will not fully commit for cell cycle. However, they will go to the upper steady state. And now I give them 24 hours. So they now choose whether they want to be here or they want to be here. And when they do the same experiment, so basically I give a 5 hours of pulse of 20%, and then I wash off my system and I come down to any lower concentration. And once I do that, I again get different distribution. But see, the nature of the distributions are different, which essentially tells you that I come down also at a different point. You can see that till point 2 also, I see a bimodal distribution earlier, which was a unimodal when I was going up. So essentially, they talk about the RBE2F's activation as happening in a switch form. And again, they prove the hypothesis that the model was proposed earlier, okay? Something like that, which was great. And eventually, this is one of the first real comprehensive model of cell cycle, which was done by Professor Goldberger's group, and Claude was a PhD student at that point of time. And this is everything said and done. This model is absolutely fantastic. The only drawback of this model is that it is based on pure Hopf bifurcation analogy. So like in some of the cases, if you go to some of the growth factor levels, this will again show some small amplitude oscillation where the proteins levels are not going up to that high level. That's the only drawback I see in this model. Otherwise, this model is absolutely very good. They have taken all possible interactions and everything. Okay, great. Now, because we didn't like that model also, so we ourselves try to make a simple deterministic model, which I will come back to my when I will give my own talk. And there we propose a kind of ODE-based model of this similar network, and we somehow created that the oscillation should come because of the saddle node invariant cycle bifurcation instead of a normal Hopf bifurcation. So this again becomes a comprehensive cell cycle model. However, here I will always get high amplitude oscillation, and my period will be much robust. It will not change with the growth factor too much. That's the whole notion for this, and nothing else. Okay, and eventually you can see that our model kind of works. So here, up till now, any questions? Because this gives more or less a synopsis or preview of the whole thing. And if you look at the mitotic phase model of whatever we talked about the frog, is basically this part of the cell cycle, mitotic to like G2M checkpoint or something like that. But here the initial part is basically my G1 phase, and then the main regulators are cyclin D and cyclin A also comes, cyclin E, and then eventually I go to the S phase where A and E plays a role, and then I come to the M phase where cyclin B plays a role, which we see in the mitotic model earlier for the frog egg extract. Any questions? Yeah. It explains many other, see, there are so many experimental papers are there. So they were not bothered about the exact, amplitude of the, how the cyclin E is going up and this. So, but there are, say, for example, Tobias Mayer's groups and other people, they are actually now measuring the exact levels of cyclin E and getting a beautiful long amplitude oscillations and everything. So there you can see that some of them are not getting, it's not matching with this model, but this model has other features are coming very nicely. And this is a very comprehensive model. So every bit of it, so Goldwater always is to detailed model. So this is a superlatively detailed model of mammalian cell cycle till now that exists in literature that I see. Our one is not that extensive, but it suffices like what we want to see. Any other question? Okay, so whatever I talked about till now is everything in terms of the deterministic model. Like deterministic means everything said and done, cells will always divide at the same, all the cells in my population will divide at the same time. It will not change. However, even when we culture cells under very healthy conditions, not all the cells divide at the same point of time. So there are some inherent fluctuations in the system or something there exist in the system which does not allow any cellular organism to behave simultaneously. If I do the experiment with the same cell even, it will show a different cell cycle time. Okay. So now the question is why we need those things. You can easily see from here the two things I will just try to illustrate that by doing a model, if I try to recreate this distribution, I will not be able to recreate this distribution just by the sake of a deterministic model. It's not possible. I cannot get a distribution of cell cycle like this. Okay, and also even at the very if I look cells growing on the culture condition, if I look at the population level or even at the lineage level, so there are very interesting phenomena that people have observed that if I think about all the cells under the life cell that are growing, I will have a distribution with some mean time and some standard deviation. And not only that, if I start following any one of these cells by saying that this is my mother cells, and then they give rise to two daughter cells, and these cells are again dividing, so I can say that these are cousin pairs. People have seen very interesting correlations that the sisters' cell cycle time are highly correlated. And the cousin cell cycle times are also highly correlated. However, the mothers and sisters cell cycle times are not at all correlated. So these sort of phenomena, which is kind of little bit mind-boggling, it's not easy to understand just only by doing a deterministic cell cycle model. There's no way it is possible. Okay. So how we can move forward to implement certain features in the cell cycle such that I will be able to accomplish these sort of data from my mathematical model? That is the question. So I have to somehow incorporate some of the fluctuating natures of the system such that I can obtain these kind of features in my model. Make sense? Okay. So this I will, again, this we should go like a story. And just to, one of the very important feature is what we call a molecular fluctuation, or the, just because we have low number of copy numbers of certain proteins and mRNAs, we have these sort of features. Just to make you understand that, we have already gone through, not those who are outside from our country, but at least within our country, we have gone through a common situation like very few years back when we have this demonetization thing. We went for our, and this is the most fortunate person who got the money, but you can see that there's a big queue, right? And whenever there was a big queue, there was, and soon you will see that this kind of a sign. And as soon as you see this kind of a sign, we are very good at creating noise. Okay, like whatever noise we can make in very good noise. Now, if I look into this characteristics of this noise, noise will be proportional to the number of people standing in the queue. Everybody will start shouting. However, in biology, we have to change our perspective. Biology also is dealing with a very similar thing. We have to change into our perspective in the sense, noise, I will not try to think with respect to how many people standing in the queue, but noise, I will try to think how many ATM machines are operative. Think about ATM machine is continuously going on and giving money, then there will be no noise. But ATM machine immediately stops and then the noise level will grow up. So biology talks about some noise, something like that. In case of biology, noise is proportional to 1 over number of ATM machines. So if the number of ATM machines are not there, I will have huge noise. Reason is, number of ATM machines are few and far. But there are many people to take the money from that ATM machine. The point here is the following. Point here is that if I look into, again, this is my cartoonish representation of a non-biologist, of a biological cell, I will have mainly few species like mRNA, proteins, genes, and this and that. If I think in terms of the protein, I will have thousands, maybe 10,000s, maybe 20,000 protein molecules at some point of time. But if I look at the corresponding mRNA, it will be 10 to 100, maybe 150, something like that. If I look at the number of copies of genes, maybe 1 and 2. So I can think that if one gene is not there, then there will be huge fluctuation. Few microRNAs or mRNAs are not there, then there will be quite a bit of fluctuation. But 1 or 2 protein molecules are not there, I will not feel that much stressed out as a cell. So biology likes this interpretation. Now, the thing is, how we know that biology likes that kind of an interpretation? People have done like absolutely great experiments with respect to, say, for example, in case of bacteria. They figured out that I can have, these proteins, these mRNAs and proteins are for genes which are just constitutively active. Means they are not getting controlled by any feedback mechanism. Just I have a mRNA and I have protein, and they are doing some jobs. They are not, regulated by any feedback mechanism. And they calculated for such kind of proteins and mRNAs, for say, for example, about 1,000 such proteins and mRNAs. They tagged all those proteins and mRNAs, and then they look at the distribution, and you can look at the distribution of the mRNA and the protein numbers, and you can simply calculate, the mean and the variance basically goes up something like that. I can do a simple birth-death process, like mRNA is getting synthesized and mRNA is getting degraded, or the protein is getting synthesized and the protein is getting degraded. I can calculate the coefficient of variation of basically the standard deviation by mean, and I can see it comes out like 1 over square root of 1 over mRNA or 1 over protein. And if I plug in the numbers, like say for example, on an average there are 3 mRNAs are there on an average, and here on an average there are 3000 protein molecules, I can easily see where the fluctuations will be more. In the mRNA I will see a huge amount of fluctuation, but in the protein I will see a very low level of fluctuation. That is not the surprising thing. The surprising thing was the following. What people find out is that these are constitutively active mRNAs and protein. So if I have low copy number of mRNA, I should have a low copy number of proteins, or if I have high copy number of proteins, I should have high copy number of mRNA. That's the normal intuition. What they figured out that there is no such correlation. I can have low copy number of mRNA, but high copy number of protein. I can have high copy number of mRNA, I can have low copy number of proteins. And then people argued that maybe you're looking at a population of cells. Each cells have different transcription factors for the same mRNA. Maybe those transcription factor concentrations are different. And that's why you are having such kind of a observation. Then comes Eloids, group. Okay. And they figured out a fantastic experiment in 2002. What they figured out is that they made bidirectional promoter, just 1 kb base pair apart. Okay. The idea is the following: one will give rise to a YFP, and then other will give you a CFP, something different. And they are under the control of the same promoter. So the idea here is that initially there is a repressor sitting on top of the promoter, and nothing is happening. I'm looking at a single cell. I'm not talking about multiple cell. Here I'm just talking about a single cell, single individual cells. In all of them I have something like this, and a repressor is sitting on top of the promoter, and nothing is happening. And then I give, to switch on, I give IPTG. As soon as I give the IPTG, my repressor is off. Now if the repressor is off and these two are 1 kb base pair apart, as a biologist I will first try to think that both these processes must kick on, that both red and green things should come up, and essentially what I should see, either of one of these, that both of them are coming out in a very coherent manner and both of them are not coming up in a coherent manner. If they come in a coherent manner, I should see the whole population as yellow. If they are not coming in an incoherent manner, I should see a mixed population. And essentially, experimentally, they observed a mixed population. Like cells are having different intensity. Now here, I cannot argue that individual cells, the transcription factors, even if they are different, for this promoter, within a cell, I am having the same concentration of the transcription factor for both the transcripts. My intuition was that if the transcription factor comes in and sit on top of this promoter, both of these should kick in. However, at any instant of time, any one of them will kick in, and that is very intrinsic to the cell. I cannot manipulate that. And that is what is essentially termed as intrinsic fluctuation or the molecular fluctuation, which is beyond our control. Make sense? This is a little bit quite complex. Yes. That is beyond our control. Let's put it like that. Even if I have, say this is a single cell, say I have this single cell, I have this thing within that. So the transcription factor that is going to sit, that concentration is constant there. Whatever may be the transcription factor, it will come and sit on top of it. And I expect that if it come and sit on top of it, I should get immediately one of them will kick in. Simultaneously, both of them will not kick. At any instant, any one of them will kick. And then obviously there are other people who will manipulate that. But I will not be able to control it in this manner. It will never happen like this. It's intrinsic property of the cells that which one will kick in, I don't know. It is completely a molecular property. At any instant of time, any one of them will fire. Any one of them will fire at any tau. Okay. Huh. Yeah, that is how way you calculate. Huh. So this is because this is very inherent. So it might happen that I have a low copy number of mRNAs, but that produces more protein because of the stability of the protein, mRNA and other things. But the other mRNA, which has a high number, maybe that is turning over very fast because of its degradation rates and other things. So there are other details of that which cannot be occupied. But the moral of the story is that at any point of time, only one of them will fire. And that is intrinsic to the system. That we cannot control. And anything else other than that will be an extrinsic factor. Like, for example, if the cell divides unequally, if the transcription factor concentrations are different, whatever may be the other things are other factors. Those are my extrinsic factors to the cell. And if it happens to be that intrinsic and extrinsic fluctuations are independent of each other, which is not often the case, if it happens, then theoretically it can be proved that the coefficient of variation square of this and the coefficient of variation square of that will be the coefficient of variation square of the total noise. So if the intrinsic and extrinsic fluctuations are independent of each other, then I will have such kind of a relation. And based on that, I can actually plot the normalized fluorescence of the green and yellow, and I should be able to calculate what is my extrinsic fluctuation contribution and what is my intrinsic fluctuation contribution by putting a bidirectional promoter. And that's people are doing this regularly in different experiments. So this is how the intrinsic molecular fluctuation and the extrinsic fluctuations are incorporated within the system. And obviously, people have, so this Thattai means our Mukund Thattai, he worked with Professor Alexander Van Oudenaarde during his stay in Princeton, I guess, if I don't. So they talked about, now we talked about all our feedbacks. So what is the role of feedback controlling these? So they have different things. So if I have a very linear system, I will have some kind of a distribution. If now I change my system with a, some kind of a negative feedback, what happens is that it reduces the fluctuations of the noise in the system. Normally, it has been observed that if I have a negative regulation, my fluctuations goes down, or if I incorporate a negative regulation. On the contrary, if I have a positive feedback regulation in a simple system, I will enhance the fluctuation. So positive feedbacks enhances the fluctuating nature of the system. Negative feedback reduces the fluctuating nature of the system, and then noise propagation can be more if you have a feed-forward kind of a system, and many other things. Those are superlative details, but essentially in cell cycle, we have all these things, and all these things controls the fluctuating nature of the cells, and eventually all this controls the fluctuating nature of the cell cycle, cell division process also. That is the whole thing. And how we can calculate that? Very simplified manner. Again, I'm not going into the details. Around 1976, numerically, there are several methods to calculate that theoretically, but one of the best numerical method was proposed around 1976, just 1 year before I born. So what it does is the following, is that if I have a multiple reactions in the system, at any instant of time, I will only care about two things. That at the next time instance, whatever maybe that time instant is, when the next reaction is going to occur. Say there are 5 different possibilities. Okay, let me show you the example, then it will be easier. Say for example, I have 3 different reactions here. 3 reactions possible. What I will first try to calculate, at what next instant of time any one of these will occur. Any one of these reactions. And in the second, what I will try to figure out is that at that time, out of these 3 reactions, which reaction will occur? Like I'm really going into the microscopic details of a system and trying to figure out this. How I do that? By calculating this joint probability. So this tau is the time when the next reaction will occur. And at that time, out of this mu reaction, one of the mueth reaction will occur. So this is how we calculate that, and there is a way to calculate it. Again, I'm not going into that details. However, if you do that, you will see that essentially when earlier, which was your particular deterministic line, you might fluctuate in and around that. Again, think it very carefully that not always that your stochastic line or the stochastic simulated line will always fall on top of your deterministic line. It will happen if you are talking, you are in a very large molecule limit. But if you are in a low number of molecules, working with low number of molecules, it might not match with your deterministic profile at all. Okay. Again, you have to somehow figure out whether the simulations are correct or not. How we utilize that? So what I just heard from some of the students that we have a little bit of talk with this model of Tyson's model. What we did, or I did with Tyson, essentially we make a bigger model. So if you go back to the previous Gillespie's thing, Gillespie's model works best or functions best if you have all the reactions in the form of mass action kinetics. So if you go back to Tyson's model, actually, this model was a phenomenological model which having Michaelis-Menten kind of a term or a Hill kind of a term and those kind of a term. So what we did, we unpacked the system in terms of the molecular mechanism. And again, we have to take things with a pinch of salt. Here is that we often model things, or say for example in this model, cyclin B was degraded by CDH1 having a Michaelis-Menten kind of a term, where CDH1 was a catalyst and cyclin B was the substrate. On the contrary, cyclin B was phosphorylating CDH1, and where cyclin B is a catalyst and CDH1 is a substrate. Now, we know in case of Michaelis-Menten, one of the very crucial assumption is that the enzyme must be in small quantity than the substrate. Now, if both of them are mutually enzyme and substrate to each other, how come both of them are small with respect to each other? So that tells you that although we use Michaelis-Menten term for in both direction and model works with charm, we know that we unpack the system in terms of molecular mechanism. We know the molecular mechanism. You unpack that system. Say earlier the system was bistable, now you will see that system bistability is gone. It's not that your model is wrong. The idea here is that you by brute force created a system which was unrealistic. Okay. However, I can get back the bistability again by fine-tuning some of the parameters, and then that is exactly what I did, and I actually also unpacked the Hill type of kinetics with molecular mechanism, and eventually we simulated the system and we got some of the features that are observed for budding yeast and fission yeast, like kind of like the what is cell cycle period, the variance and everything, with incorporating intrinsic molecular fluctuation and also the extrinsic molecular fluctuation that we talked about, that the unequal division, these, that, everything was there in that model. And we can figure out that many experimental data we can reconcile even with low copy number of mRNAs and everything from our simulation. We can calculate all these features from our simulation, okay, at that stage. However, as I showed you earlier, that these were Yao et al.'s paper, and then eventually in my lab earlier, we try to do a very similar kind of a pulse kind of an experiment and try to get, can I simulate at least qualitatively the way they have these sort of qualitative experimental observation, that can I simulate a system with a restriction point control mechanism if I do a pulse up and without pulse experiments numerically, and I have 10,000 cells and I take the distribution, I can somehow create the distribution and talk about some very intricate features of these models. Yes. Then it will be with that model itself. Yeah, you can do that. Is what we used to call a poor man's Gillespie. That we cannot do anything. I do a Gillespie of that. It will also give you a result, but that will not be reliable in most of the cases.
So, I mean, unpacking it and doing Gillespie.
Most of the time it is not. Most of the time it is next to impossible.
Yeah, but if you are able to
That will be ideal. That will be the ideal thing, because
versus not unpacked, these two may not give exactly same results.
yes, yes, absolutely.
So ideally you want to unpack it and then do the Gillespie, because Gillespie was built for these
simple things. Yes, yes. Yeah. Yes. Exactly. Yes, but but the problem is to get back that situation. It is not possible because in your actual model you assume both the pairs of protein as their mutual Michaelis-Menten enzyme kinetics pair, which is impossible to, you know. Then it will match.
But most of the time it will
not match. Okay, with this slide, I just want to finish, and I think I'm perfectly on time. That why we need further models then? We already have so many models and this and that, right? So the point here is that although we have certain models, now the experimental procedure has gone up to a certain level, and our sophistication in the model should go up to that level also. We should be able to capture the cell type specificity. I should not say this is a mammalian cell cycle model. I should say this will be a model for HeLa cells. This will be a model for MCF7 or something like that. I should have a cell type specific cell cycle model. And to do that, I have to capture many features, not only the simple cell cycle periods or something. There are many other features also. We have to capture the how the different fluctuations are governing the system. Is not trivial because there are many factors are there which we are trying to incorporate in our cell cycle model. Not only that, at the end of the day, we want to go from this cell cycle model to somehow capture how this will relate to the ultimate phenotypic outcome. Should it be like proliferation will be very high, or the migration will be very high, or something like that? Can I somehow correlate some output of the cell cycle with these phenotypic outcomes? So these are some of the challenges that I will try to showcase what we are trying to do in our lab based on whatever cell cycle knowledge I try to impart a little bit, although little hand waving, but this will make you kickstart. And if you really want to move on to something like that, there are beautiful literature. You can go and study and learn these things. This is something that what I will say is that this is the way the science of the systems biology of cell cycle has progressed is a mathematical modeller's charm, because this is a true sense. A mathematical model was proposed and experimentally was later validated. Normally, and I'm saying this as a mathematical modeler plus an experimental scientist, is that sometimes we get an experiment, we try to prove this, and we make a model. That is one way of doing it. There, the intuitions were created, and intuitions were built up, and then the intuitions were fall in place at the later, and that is something, the beauty of the cell cycle dynamics. That is what I think, and with this I want to end this part. Is any questions if you have? Yeah, please. This is an important thing. Getting the credit or not credit cannot be assured. Let's put it like that. Because sometimes what happens is that, unfortunately, still in our scientific community, there exists, we can see that people have seen the paper, people have learned the paper, people have asked the paper from me, and then they don't refer, then refer another paper. This has happened. But you cannot do science by thinking about this. If your work is good, it will get recognized. That is my feeling. And although James Farrell and Tyson is not in great terms, but they mutually admire each other. That I know. So it's fine. I don't see anything wrong in it. Somebody else developed the same model in 51, identical, not referred to Langmuir, but today also the model goes by the name of the person who developed it. Just like that. Questions? Any other? Please feel free to ask any question. Nothing is.
You say that there is some correlation in cell cycle time. You also see some correlation in the volume of the cells over the phases because
Yes. Yes, I will. I think Mark Krishna's group have tried a bit on cell size. No, I don't know whether in terms of cell volume or not. The point is that to how we measure that very scrupulously as a function of time, that is not always easy. But if it is done, for example, it is well known in budding yeast, there is a very good correlation that if the cell grows very big, if the mother cell grows very big, then it divides very quickly and then divides into small size and then it grows again bigger. So there is a very tight size control mechanism in budding yeast cell cycle. And then, but for the mammalian cell cycle, there is a size control mechanism, but it is not as superlatively active like a budding yeast. Yeah, 100%. The population doubling by exactly the cell cycle time of that. So basically, there is a definitive correlation. So there are some correlation graphs also. For budding yeast, so there is definite relations. Yes.
For mammalian cells, it's easy to measure the area, right? So area, there are strong correlations.
Strong correlations. So he will know. Sorry about that. We will come to that. We will come to that in the second part. See, these are all premises. So I created so that you don't fall suddenly what is coming on. This is all intro. This is all intro. We will come. I hope something like is there. I hope. It will take time. It will take time. See, quantifying all these things experimentally is not easy. But nowadays, maybe it will.
So I haven't followed the literature much, but I've noticed that there are now debates about the restriction point. People are saying, like you said, right, toward the end of G1 phase, we used to think is the restriction point. But now I think a lot of people are saying that, including, they are saying that that's no longer where the restriction point is. They are saying it's somewhere else. Have you followed this?
Yes. Recently there was a paper. Yeah, I haven't followed it deeply. I have seen that. Yes. Okay, and then there's how the cells can come back from different places. Have you seen that? Right. So we were also trying. So there was I don't know whether there's a cell system paper where they talked about that how if you give them different stress to cells, different kinds of stress like serum stress, like anything else, where the cells will get stuck in the cell cycle, and that was very interesting. And we were also actually working with Janwa with another project, like during the EMT, where the cells get stuck and why they get stuck there. We were working with hypoxia, And we also see that if you give them hypoxic condition, cells have a unique way of, combat that. I will tell you that. That was amazing. And I don't know why it is happening. And we have seen that in two different cell lines. Maybe I will not talk today, but yes. Yes, yes, yes. Yes. Yes, their goal. If you look at Goldbetter paper, there is a very extensive paper of Goldbetter on how circadian clock is coupled with cell cycle, and there's a connection via V1 and what was the other one? Cyclin B and something, and there are some circadian regulators and how they connect with the cell cycle. We were thinking about figuring. So there was, I will come to the point where this mother-daughter correlation and daughter-daughter correlation, that thing. So there was a paper in 2015 from the Natalia Balaban's group, and they showed that because this happens because cell cycle was kind of influenced by circadian clocks of those cells, and if they come into similar phases, then only the daughter cells gets correlated or something like that. So that was one of them. But we tried with but their model was kind of a, what can I say, more statistical model. We tried with our network-based model, but I haven't been able to reproduce the correlation yet. So I'm not 100% that how it is coming. So I have to see. We are trying. Huh? Yes.
So we can get the correlations.
Yes, what?
I can show you.
Okay. With the circadian? Okay. Fine. That's great. We will see. Any other questions?